Hey everybody. This little talk is going to be about understanding the neurons, and what I'm going to do is go quickly through the concepts of how neurons inside our head work and how people have mathematically tried to create artificial neural network models, those of McCulloch and Pitts, VLSI, spiking neural networks, to name a few. So this little intro video is going to just cover a few concepts. So let's get started. So, there are about 10 to the power 11 neurons in our brain, and almost 10 to the power 10 are in the cortex. No wonder, neocortex has been a super interesting area for researchers for the last century or so. The cortex is the outer mantle of cells surrounding the central structures, such as the brainstem and the thalamus. Now the cortices are about 2 to 3 millimeter thick in the humans, and is folded on an average size of a surface area of about 2.2 to 2.2 meter squares. The neuron, which is essentially a nerve cell, is mostly the most important functional unit of a central nervous system, including a brain. A neuron is basically an extension of a simple cell with two types of appendages. So you have multiple dendrites and you have the axon. Just like you can see here, for example, here you have this is essentially your soma, the cell body. Your nucleus is here, mitochondrion. This is your axon, and these are your dendrites. And basically, so that's a structure. A neuron actually possesses all the internal structures of a regular cell, comprising of 4 components, which is your dendrites, I just showed you, the soma, the cell body with the cell nucleus, the axon, and obviously the synapses. So a soma contains a cell nucleus. The dendrites basically branch out into a bushy network around the cell to receive input from other neurons, whereas the axon stretches over a long distance. Typically, sometimes it's a centimeter, and it can extend to a meter as well in extreme cases inside your brain. Actually, that's interesting, isn't it? The axon is the output channel to other neurons. It branches into strands and substrands to connect to the dendrites and cell bodies of other neurons. Now, connecting junction is actually called a synapse, which you saw in the picture briefly. Each cortical neuron actually receives about 100 to 105 synaptic connections, with most inputs coming from distant neurons. Therefore, the connection in the cortex are said to exhibit long-range excitation and short-range inhibition. A neuron receives signals from other neurons through its soma and dendrites. It integrates them, sends the output signals to other neurons through its axon. The dendrites receive signals from other neighboring neurons and pass these on to the cell body, and are processed therein, and resulting signals are transferred through an axon. Like any other cell, a neuron has a membrane potential. I think I've spoken about that a little bit in India as well last time. That is electrical potential difference between the intracellular and extracellular compartments caused by the different densities of sodium and potassium. The neuronal membrane has relatively selective ionic channels that allow some specific ions to cross the membrane. The cell membrane has electric potential, which is the resting potential, of about minus 70 millivoltage, which is maintained by pumping positive ions, which is Na plus, out of the cell. Now, unlike an ordinary cell, and as you know, our whole body is comprised of cells, about 3.7 trillion. If you ask, physicians, experienced people, the neurons is actually excitable. This is not excitable. The skin is not, for example, the cells comprising of my skin, not. Because of the inputs from the dendrites, the cell may not be able to maintain the minus 70 millivolt resting potential, resulting into an action potential that is pulse transmitted down the axon. the image that you saw. The signals are propagated from neuron to neuron by rather complicated electrochemical reaction. So the chemical transmitters, substances actually pass the synapses and enter the dendrites, changing the electric potential of the cell body. When the potential is above a certain threshold, the electric pulse, or an action potential, is sent along the axon. After releasing the pulse, the neurons returns to its resting potential. So remember, resting potential is about minus 70 MV, and there is an action potential which is the changed value. The action potential causes the release of certain biochemical agents for transmitting messages or to the dendrites of nearby neurons. These biochemical transmitters may have either an excitatory or inhibitory effects on the neighboring neurons. A synapse that increases the potential of excitatory, whereas a synapse that decreases is inhibitory. Synaptic connections exhibit plasticity. What does that mean? It's basically long-term change in the strength of the connections in response to the pattern of stimulation. That's what plasticity is. So neurons also form new connections from other neurons, and sometimes an entire collection of neurons can migrate from one place to another. So that's kind of interesting, isn't it? These mechanisms are thought to kind of form the basis of learning of the brain, you know? Synaptic plasticity is basic biological mechanism underlying the learning and memory. So your hippocampus here and the learning, I guess, throughout a bunch of other things, it's a long topic to discuss here. So two types of plasticity here is Hebbian plasticity, and there is a compensatory homeostatic plasticity, which stabilizes neuronal activity, for example. So there are two types of plasticity. So inspired by this, a large number of learning rules specifying how activity and training experiences change the synaptic efficacies have been advanced. Physiological neurons are typically either excitatory or inhibitory, but they cannot be both. Well, people would argue they are. So the principle states that neuronal activity releases some transmitters at all branches of their axons. Although there are exceptions, most neurons are exclusively excitatory or inhibitory most of the time. A mixture of excitatory and inhibitory functional connections could be realized by a purely excitatory projection in parallel, for example, with a 2-synapse projection through an inhibitory population. This works well with the ratios of excitatory and inhibitory that are realistic for the neocortex. Now, mixed excitatory and inhibitory functional connections can be realized in networks that are dominated by inhibition, such as those in the basal ganglia. So basically, I spoke about a lot of things. We spoke about the neurons, explained to you a little bit about how a neuron is different, is essentially very, very biochemically different cell than your neural, which is in your deep learning. The reason why we believe, or I believe at least, and I'm learning myself all the time about it, why it's important is that we still believe that the deep learning models have been super successful, but they haven't actually reached that potential of becoming biologically plausible, like you and I are, how we function. So I really think that diving into this area is going to help us actually develop better, not only better models and algorithms, but also potentially better products when we actually get into a brain-machine interface. So there are a bunch of project ideas we're discussing with partners to develop maybe at some point in time as a brain-machine interface project. And I'm very happy to sort of talk about this. Do see a prolongated sort of discussion into our groups channel. What I will do is I will also go into talking about Hebbian or a McCulloch's and Pitts model, a VLSI model, maybe spiking neural network model as well. Kind of we'll sketch these, we'll draw these, and try to understand what these are. So looking into the past is a way for me to actually understand the future. And obviously we'll also start doing some more interesting things, and I'll look forward to seeing you guys there. So take care.